I will start in 2-3 minutes. I'll start in just 2 minutes. Right, so I'll share my slides. So this is the 7th live session for this course. I've uploaded the videos of the previous 6 live sessions and shared the links. So you can always go through them. And as always, this session is will also be recorded and uploaded. So in this session, as always, I'll firstly cover the videos of week 7 that Sir had gone through. So I'll be going through each of the videos in short. And then we'll see some practice problems of the week 6 assignment. So I'll up to the previous 2 classes around, it was all about nucleic acids and NMR of nucleic acids. From this class, we'll start looking at proteins. So protein is a polymer, the monomer of which is called an amino acid. And in an amino acid, you have an NH2 group, a COOH group. So the NH2 is the N terminus, the COOH or carboxylic acid group is the C terminus. And the carbon to which both is attached is called the alpha carbon. And to the alpha carbon, you have one hydrogen and another R group. Now, this R group can be of 20 different types, forming the 20 different amino acids. And when 2 amino acids combine with each other, a dehydration reaction takes place, that is, a molecule of water is lost. So one H from an amino group and the OH from the carboxylic acid of another amino acid is lost as a molecule of water, and a new bond, the CONH bond, is formed, which is an amide bond or a peptide bond. So from two amino acids, you can get a dipeptide. And like this, hundreds and thousands of amino acids can combine to form a long peptide chain, which eventually folds into the protein. Now, these amino acids, since this alpha carbon is attached to 4 different groups, it is chiral in nature, which means that the mirror image of the amino acid is non-superimposable with each other. So each amino acid can have 2 different configurations known as the L and the D configuration. Now, normally in nature, the L amino acid is the one which is naturally occurring. So all the proteins in your body or in the food that we eat, they are made up of L amino acids. And if we want a D amino acid, it will have to be synthetically made. Because they are 2 different enantiomers, they cannot be interconverted into one another. Now we will briefly look into the 20 different amino acids that exist in nature. So this R group can be charged or neutral. So first we will look at the cases where the, just one second, I think someone wants to join. Yeah, so I was just going through the different R groups of the amino acids. So there are 20 amino acids in total, and there can be 20 different R groups. Now, this R group can be charged, and under the charge, there can be 2 kinds of charges, positive or negative charges. Under the positive charge, you have 3 amino acids, arginine, histidine, and lysine. So you can see this region is common in all 3. What is different is the R group. That is why the name amino acid is common, because in each you have an amino group and a carboxylic acid group. Now the side chain is different. In lysine, you have a long aliphatic CH2-CH2 chain ending with an NH2 group. In histidine, you have a CH2 attached to an aromatic imidazole ring. And in arginine, you have again a long CH2-CH2 chain attached to this kind of a group, which is called a guanidinium group. Now, the common thing between these 3 is that the side chain has a basic group. So, as you can see, NH2 of lysine can catch a H plus and act as a base. Similarly, this N of histidine or this N can donate a lone pair to a H plus and act as a base, and same for arginine. So when they accept an H plus, they get positively charged, and they also act as Lewis bases because they are donating a lone pair. So these are basic amino acids having positively charged side chains. There are 2 amino acids which fall under the negatively charged side chains. Here you can see is aspartic acid having a side chain of CH2COOH, and glutamic acid has one extra CH2 here, and again a COOH group. Now in neutral conditions, the COOH group, it loses, so it exists as COOH. This is lost as H plus, and it exists as a negatively charged side chain COO minus. That is why this falls under the negatively charged amino acids, or acidic, because it is donating a H plus, and so it is acting as an acidic amino acid. Then you can have amino acids, the other amino acids all have neutral side chains. Now within the neutral also, there are certain categories. One is where the side chain is neutral, but there are polar residues. Like if you look at serine, it has a side chain CH2OH. So O is an electronegative molecule. So O will have a partial negative charge. This CH2 will have a partial positive charge. So it is polar, but it does not have a full charge like this. These are only partial charges. So we can write this as delta minus and delta plus. But overall, the molecule is neutral. Similarly, you can have threonine, which is like serine, but there's an extra methyl group here. And asparagine and glutamine are where the side chains have an amide group. So in asparagine, it is CH2, CONH2, and glutamine has one extra CH2 here, and again ends in CONH2. So these four, they have polar residues like oxygen or nitrogen, but they do not have a charge like the acidic and basic amino acids. They are overall still neutral. Then you have some special cases of amino acids. And in this, why they are special is, first of all, we look at them one by one. Cysteine is special because it has a sulfur atom. Sometimes I'll just reduce the size of this. The lower parts are not being seen. So, yeah. So cysteine is special because it has a CH2SH group. So it has a sulfur group, and the special thing about cysteine is that two cysteine molecules can come close together. They can lose, so two cysteine molecules, and they can form a disulfide bond. They will both will lose this hydrogen and they will form a disulfide bond. Then you have glycine, where the R group is another H. So glycine is the only amino acid where this alpha carbon is not chiral. Because for it to be chiral, you need to have four different groups attached to it. But in glycine, both the groups on the alpha carbon are hydrogen. Then proline is another special amino acid because this NH is a secondary amine. So in all the other amino acids, if you see, it is NH2. That is called a primary amine. But proline, the backbone forms a cyclic, 5-membered cyclic ring. So proline, the NH2 actually becomes NH. So when this proline forms a dipeptide, this hydrogen is lost, and then there is no amide hydrogen on proline. Because when it's forming a dipeptide, imagine it is connecting with the CO group of another amino acid, then this hydrogen is lost. So when proline exists in a peptide chain, the amide nitrogen will not have any hydrogen attached to it. And the last class of amino acid is where they are neutral, but the side chain is hydrophobic. Hydrophobic meaning they are mostly like long aliphatic chains. So it does not need to be long, but it's an aliphatic chain. So in this, you have alanine, where the R is just a CH2 group, CH3 group. You have valine, where R is a isopropyl group, CH attached to two CH3s. Then you have isoleucine, where again you can see it is a secondary butyl group. Leucine, where it is an isobutyryl group. So these four, the R group is only consists of carbon and hydrogen, and they can be branched in different ways. Then you have methionine, where it is again different CH2, CH3 groups, but in the middle you have one sulfur. So this can sometimes fall within the polar uncharged side. So in some cases, it is classified as a polar uncharged side chain also. And because it has so many CH2 groups, it can fall inside the hydrophobic side chain also. The other three amino acids basically have aromatic side chains. So histidine also, if you remember, this ring is aromatic. But because it could accept one H plus, it falls within the charged basic side chains. Phenylalanine, it just has a CH2PH group. So there is no question of any charge here. It's just an aromatic side chain. Tyrosine is the same as phenylalanine, but there is an OH group at the para position. So you have this, the main backbone of the amino acid, and at the para position, you have an OH group. And finally, there is tryptophan, where you have this kind of an indole ring attached at the R group. So these three form are part of the hydrophobic side chain amino acids. Now, you can think that OH or NH, they can be polar, but because of the large aromatic residues, their uh, characteristic is more hydrophobic compared to polar. That is why they are classified under the hydrophobic side chain amino acids. So in this way, by having 20 different R groups, you can have 20 different naturally occurring amino acids. Now, each amino acid has their 3-letter code and their 1-letter code, which is shown here, which is something generally as you do the NMR of proteins, you kind of have to remember, because the sequence, in the sequence, the whole name won't be written. Either the 3-letter or the 1-letter code will be written. So those are the basic components that make up the protein. Now we look at the different structures of the protein. So the primary structure is the one that is made up of a long chain of amino acids, and it is just the sequence of the chain. So if you know the sequence of the chain, like suppose glycine, proline, asparagine, here if you consider each bead as an amino acid, then this entire sequence of amino acids, once you have determined, then you know the primary protein structure. This structure is folded in different ways. It can form an alpha helix, or it can form a sheet-like molecule. We'll look into the details of this in just some time. So then it is called a secondary structure. Basically, in secondary structure, there is local folding of the polypeptide chain. So if you look, this is the alpha helix. In the alpha helix, there are hydrogen bonds. So there is hydrogen bond between CO and NH of I+4 and I residues. So if I consider this as the NH of I, if I consider this as the NH of I, then this is the NH of I+1. This is I+2. This is I+3. And this carbonyl, which is forming the hydrogen bond here, this carbonyl is I+4. So I'll again discard this. So this hydrogen bond that is formed between the CO and NH is between the I+4 residues CO and the I residues NH. This kind of repeated hydrogen bond forms between I+4 and I residue, and that leads to the formation of the alpha helix. So so in secondary protein structures, basically there is repetition of local interactions happening and a kind of ordering of structure happens. Another kind of secondary structure is the beta sheet. So here you have only one polypeptide chain. In the beta sheet, you have two chains which can either be parallel or anti-parallel to each other. So in this case, the top chain is said to be N to C, because if you consider, we will ignore the first hydrogen bond. From the second hydrogen bond, if we consider, it goes from N, C, CO, N, C, CO. So it starts from N, goes to the C terminal. And in case of the below chain, again, we will ignore the first hydrogen bond. It is CO, NC, CO, NC. So it starts from the C terminal, that is the carboxyl terminal, and goes to the N terminal. So it is said to be N to C. So the direction of the two chains are opposite, and so they are said to be anti-parallel beta sheets. Now, between the two strands, there are hydrogen bonds between the CO and NH. So again, here there are regular hydrogen bonds holding the polypeptide chains together in a kind of regular structure. And these two are different kinds of secondary structures. There can be many other kinds of secondary structures. We'll look into that in some time. Then what happens is that there is a three-dimensional folding of a protein due to side chain interactions, and that leads to tertiary structure. So imagine you have a protein like this. In the protein, some segments are forming alpha helix, some segments are forming beta sheet, but the entire protein, you can see here, this section shown here is a alpha helix. This section shown here is a beta sheet. But the entire three-dimensional structure is said to be a tertiary structure. And here there can be many other interactions like van der Waals interactions, hydrophobic pi-pi interactions, and so on. So secondary structures are just parts of the tertiary structure which are regular. But the tertiary structure as a whole does not have any regularity, and it is the entire three-dimensional structure of a protein. So you can think of it as relative orientation of different secondary structures. And finally, you have the quaternary structure, where if you have a protein consisting of more than one amino acid chain. So you can see here, the blue is one amino acid chain, and this orange here is another amino acid chain. The two chains can be held together by different interactions such as hydrogen bonding, van der Waals, and so on. And then that is called a quaternary structure. So these are basically the levels of organization in a protein. Now we will look at another kind of tertiary secondary structure. So in the previous slide, we looked at the beta sheet and the alpha helix. Now this is another kind of secondary structure called the turn. So if you look at the tertiary structure here, the two strands form a beta sheet, but between one strand and the next, which is highlighted here, there is a turn. This kind of turn is basically formed by 4 amino acid residues shown here. So this is the general structure of a turn. You have, so the amino acids are labeled. So this is the ith amino acid residue. So each amino acid consists of N, C alpha, CO. In the ith amino acid residue, the N is not shown. So imagine there's an N here, C alpha, CO, that is I. N C alpha CO, that is I plus 1. Again, N C alpha CO, that is I plus 2. And N C alpha, again, CO is not shown. Imagine there's a CO here, that is I plus 3. So total of 4 amino acids make up the turn. And in the turn, there is an intramolecular hydrogen bonding between the CO of the I residue and the NH of the I plus third residue. And normally in a turn, these two strands come close enough that the C alpha of I and C alpha of I plus 3 is within 7 angstrom of each other. Now these turns are generally present wherever there is a change in direction of the chain. So you have the polypeptide chain folding in different ways, and wherever you have a change in the direction of the chain, there, so you can see here the upper blue chain is going from left to right, and the lower blue chain is going from right to left. So there's a change of direction, and wherever there is such a change of direction, there is a turn. The turns can also be of different types depending on what are the torsional angle residues. So we will look at the different torsion angles in some time. But you can see here there are two types of turns shown. So the first residue is the same in both the turns. In the second residue, this carboxyl is pointing down and this carboxyl is pointing up. So this is a main change. Another thing you can think is that both the H and R group of the I plus first residue is outwards, whereas here R is outwards, H is inwards. Similarly, there are certain small changes in the orientation of the different amino acids, and that is how different kinds of the turns are formed. These are often called beta turns because most often they are found in beta sheets. So that is, so this also forms another kind of secondary structure. Now we will look a little bit into more detail of the beta sheet. We have already seen the anti-parallel beta sheet. We are just showing that again here. So the top strand, it starts from N and goes to C. So you can see this is an N to C strand. The second strand, it starts from CO and goes to N. So it is a C to N strand. So between first and second strand, an anti-parallel beta sheet is formed because the two strands are in opposite direction. The third strand is again N to C. So N to C, and between second and third strand, they are again in opposite directions. So between second and third also, it is an anti-parallel beta sheet. The fourth strand is again NH to CO, that is N to C. So between third and fourth strand, both are in the same direction. You can see NH here, NH here, CO here, CO here. So the atoms are same between the two strands. So because they are in the same direction, they are called parallel beta sheets. Now the pattern of hydrogen bonding is slightly different in antiparallel and parallel beta sheet. In antiparallel, you can see the NH and CO are directly facing each other. Again here CO and NH directly facing each other. So the hydrogen bonds are straight and so they are much stronger. Whereas in this parallel beta sheet, NH and CO are at an angle to each other. So the hydrogen bond is weaker because the distance between H and O is also greater. So between parallel and antiparallel, antiparallel is the more stable beta sheet that forms. And you can see there are cyclic structures formed due to these hydrogen bonds. So if we see the antiparallel beta sheet, we'll start counting from this carbon as 1. So 1, 2, 3, 4, 5, 6, 7, 8, 9, then this atom becomes 10. So this entire thing is a 10-membered cyclic structure. So it's written as 10. Similarly, if you count the number of atoms in the next cyclic structure, you will see 14. So in antiparallel beta sheet, two kinds of cyclic hydrogen bonded structures are found, alternating 10 and 14 membered cyclic structures. You can see it is better illustrated in this antiparallel beta sheet. This is a 10-membered ring, this is a 14-membered ring. Again 10, again 14. So the pattern of hydrogen bonding is different, and that can often help to distinguish between the parallel and antiparallel beta sheet in NMR. In parallel beta sheet, on the other hand, if we count the number of atoms starting from O, it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So a 12-membered cyclic structure is formed, and you can see it is uniform everywhere. So all the cyclic structures formed in the parallel beta sheet are 12-membered. So that is another difference. And this is all about the beta sheet. So now we will look at the dihedral angles like I was talking about. So this is a peptide chain that is shown. You have NH-CO, C-alpha. Again, NH-CO, C-alpha. So here a peptide chain is shown. Now there are three kinds of dihedral angles that are important. The first dihedral angle, so you know dihedral angle is made up of 4 atoms. So the first dihedral angle starts from C alpha, N, C. Okay, so the C double bond O is often called C prime. So I'll just label this. This is often called C dash or C prime. So C alpha, N, C prime, C alpha, that forms the omega dihedral angle. So basically rotation along the N-C prime bond is called omega. This omega can be of two types. Firstly, you can have an orientation where the omega is 180. So the C alpha and C alpha are on opposite sides. Here the C alpha is up, here the C alpha is down. So this is one orientation, and it is called trans, because if you imagine a double bond between C and N, the two C alphas are on opposite sides, so it's trans. Or you can have a cis orientation where the omega angle is 0. So C alpha, C alpha is 0, and both the C alphas are facing each other. So this is a much more unstable structure because there is steric repulsion between the C alpha and the substituents on C alpha. So generally the trans orientation is favored for most peptide bonds, where because the two C alphas are on opposite direction, this kind of steric problems do not exist. Now, why can't 180 and 0 just naturally convert into each other? That cannot happen because this kind of resonance. So in the amide N, there is a lone pair, and the lone pair can delocalize into the carbonyl C double bond O. So this is one resonance structure where you have the lone pair donated into the carbonyl. So the carbonyl has CO minus and this NH has a plus. So the amide bond or peptide bond exists somewhere between these two resonance structures. So the amide bond has a partial double bond structure. And due to this partial double bond structure, the rotation along the amide bond is not free. So the omega 0 or the cis orientation cannot naturally interconvert into the trans orientation because of this partial double bond character. Another effect of this partial double bond character is that it causes the peptide bond to be planar. So you can see different planes shown here in this color. Each of these planes have a peptide bond. So you can see this NC is the peptide bond, and this bond has to be planar because of this partial double bond character. So each amino acid lies in a plane which we are calling as the peptide plane. There are two other torsion angles which are important. One is starting from N, C prime, C alpha, N. That is called the Chi torsion angle, shown in red here. And another is starting from C prime, C alpha, N, C prime. That is the Phi torsion angle shown here. So these two torsion angles, they are also, it is not like we can have any value of these torsion angles from 0 to 360 degree, because you can see an example shown here. So we saw that phi is from N to the next N. So here also it's N, C alpha, C prime, N. So this rotation angle is phi. Now you can see that at the angle phi shown here, there will be steric repulsion between these two oxygens. So this is not a stable orientation of phi. So these are called disallowed torsions, where the steric clashes are too high, and so the molecule cannot exist in those torsions. So Ramachandran had simulated different torsion angles, and he had given a plot of phi versus Chi, where most of the plot is composed of disallowed torsions. So all the gray colored regions of this plot are places where this kind of steric clashes happen, and so that torsion is not allowed. The dark red regions shown here are the regions which are highly favored, that is, least steric clashes happen. And the light red regions are basically where you have some amount of steric clashes. So they are less allowed, but still the overall stability is there. So some conformations do exist in the light red regions. So they are often called as partially allowed regions. So within this, this Ramachandran plot basically shows you what are the allowed phi Chi torsions in any protein that you can have. And you can see different regions of the Ramachandran map are actually belonging to different secondary structures. So if you have a region here, which is shown by this red dot, which has a phi Chi value of around minus 60, and here also it's around minus 60. So that would form your alpha helix or right-handed alpha helix. Then you can have a beta sheet and parallel and anti-parallel beta sheets. You can see they occurred in different regions, but in both the regions, the Chi is positive and the phi is negative. So like this, different regions of the Ramachandran, so different secondary structures occupy different regions of the Ramachandran map. So now we will slowly start coming into the NMR aspects of these proteins. So here a 1H spectrum is shown of a folded versus an unfolded protein. So you can imagine a protein like this, which is in the folded state. And the folded state can have many different kinds of secondary structures. There are helices shown here, sheets shown here, and so on. If you heat this protein or give some other kind of chemical in order to break the peptide bonds, it can unfold into into the primary structure of the protein, which is just a polypeptide chain. So what will be the difference in NMR here? As you can see, the folded structure has many distinct peaks. But in the unfolded structure, the peaks are all kind of getting merged into a narrow range. So you can see here in the folded structure, the range of peaks here is this much. But in unfolded structure, mostly the range has reduced. There is one peak here, but otherwise the range has reduced. So why is that? Basically, the unfolded structure is a very dynamic structure, meaning that it can exist. So this is one unfolded structure. You can have another unfolded structure, something like this. The point is that there is not one unique unfolded structure. The unfolded state can exist in many different structures. So it's a very dynamic molecule. That is, multiple conformations are possible for each amino acid. Because of that, since so many conformations are possible, all the amino acids kind of merge into a narrow range. And that is the basic difference between your folded and unfolded protein NMR. Now, in the NMR, like in DNA, if you remember, we had certain regions where the sugar protons are expected, certain regions where the base protons are expected, and so on. Even in protein, there are certain regions corresponding to different groups. So around 10.5 to around 6.8 or so, you have the backbone amide protons, which are shown here as, so backbone amide proton is the ones which are part of the amide bond. So CONH, that's part of the amide bond, CONH, and so on. Then you have an aromatic region from around 6.8 to 8. Aromatic proton are basically, you have, while discussing the amino acids, we discuss the ones which have aromatic side chain. Here phenylalanine is shown. So the phenylalanine protons will appear in this region. Then you have another region around 6 to 7.6 or so on, where the side chain amide groups appear. So which are the side chain amide groups? We know asparagine and glutamine. Asparagine and glutamine are the ones which have side chain amide. So side chain CONH2, side chain CONH2. This is the region where they appear. Then you have a broad region from around 3.5 to 5.6 or so on, where the H alpha appears. And lower than that, you will get the different aliphatic protons or methyl protons. So there are characteristic ranges in which the different protons appear. Now, there are certain namings in in the amino acid side chain, there is a certain nomenclature of the protons of the side chains. We will briefly go into that. So we'll start with the simplest, that is glycine. So the C alpha has two hydrogens. Here they have just been written as H alpha 2 and H alpha 3. Then if you have, so glycine does not have any other side chain other than hydrogen. If you have something like alanine, then the carbon attached to C alpha is the C beta. And the hydrogens attached to C beta are also called H beta. If you have longer side chains, like suppose leucine, so in leucine you have a C alpha, which is, you have a C alpha, which is attached to C beta, which is attached to C gamma, which is then attached to C delta. So that is how the nomenclature goes. If you have an even longer side chain, so we look at something like lysine. In lysine, you have C, again, imagine there's a C alpha here, then C alpha, then C beta, gamma, delta, epsilon, and then after epsilon, it's eta. So the nitrogen has been numbered as eta. That is how the numbering basically goes for different amino acids. And so you can, in the aromatic amino acids, if you look, we look at phenylalanine. We have C alpha attached to C beta, attached to C gamma. Then the two carbons attached to C gamma have been labeled as delta 1, delta 2. The two carbons after that are epsilon 1 and epsilon 2. And the carbon after that is the eta. So after epsilon, you have eta. And the nomenclature of the hydrogen is basically the same as the carbon. If there are two hydrogens, you can label them as delta 1, delta 2, and so on. So that is how the different atoms are labeled. This is important when we are doing NMR, because as we'll discuss the different spectra, we will talk about interactions between different hydrogens, and the hydrogens will be named like this. So that is why I've just briefly gone into this. So as you can see here, we talked about the ranges at which the different protons appear. This is more details into that. So you have the 20 amino acids. You can see that the H alpha of most of the amino acids lie around 5.4 to around 4. So that is where the H alpha lies. Then more upfield, you can have the H beta and so on. Of course, there are certain exceptions to this depending on where the polar group is attached. So we look at alanine, which is the first one. You can see alpha and beta chemical shifts are well separated. So why is there a range of chemical shift? Because depending on the sequence of the polypeptide, if suppose alanine is next to a acidic amino acid, then as that acidic amino acid will have a charged side chain, and that charged side chain will have some effect on the chemical shift of alanine. That is why there's always a range. So in alanine, the alpha and beta are well differentiated. But if you go to something like serine, serine is basically alanine, but to the CH3 group, one OH is attached. Now OH will have an electron withdrawing effect. So the CH2 will get a delta plus charge. So in serine, you can see the beta. So beta in alanine was here. Beta in serine is here. It has moved downfield. Because of this electron withdrawing effect and the delta plus charge that has developed, the beta, so this is the beta hydrogen. This is the beta hydrogen. Beta hydrogen is becoming more downfield shifted in serine. So in serine, alpha and beta are actually very close to each other. Similarly, you can have threonine. In threonine, there is an extra methyl group. So I'll just remove this. You can see threonine is basically serine, but in the beta carbon, there is an extra methyl group. So threonine has a C gamma also. This is alpha, this is beta, and this is gamma. So threonine has, if you look at threonine, it has alpha, beta, and gamma. So alpha and beta in threonine, similar to serine, are very close to each other because of the electron withdrawing effect of the OH group. But gamma is again pretty upfield because gamma hydrogen is not directly connected to the OH group. So like that, different amino acids can have different ranges of the shifts. So this is the basic 1D NMR spectra. Then you can have your 2D NMR spectra, where the toxi spectra is mostly used for spin system identification. What does that mean? Spin system identification basically means that you're identifying the particular amino acid. So using toxi and cosi spectra, we can find out that what is the sequence of amino acid there is. we will go into more details a bit later. This is just a basic toxi spectra that is shown here. Each vertical line is basically corresponding to the same residue. So suppose we have leucine. In leucine, you have the NH amide protons. These are connected to the alpha, beta, gamma, delta, and so on protons. So in leucine, you expect up to H delta. So indeed, you can see these are, one of this is alpha, one is beta, and then you have other peaks here, which are the delta peak. So delta peak in leucine is directly, leucine, the delta is here. So delta will be somewhere here, and then you have the amide. Here it appears as two peaks, but it is actually here basically your gamma and delta are combined. And then you have your NH protons. So similarly, along each vertical line, you basically have one amino acid. Now, in some cases, in the NH region, you can have two protons. That is where basically you have, this is the NH side chain region. Normally in the NH side chain region, you will only have protons if the amino acid is asparagine or glutamine. But so in leucine, you can see there is no peak in this region, which is the NH side chain region. So these that are shown here, even in the lysine also has NH side chain. So lysine side chain, if you look at it, it has this is charged lysine. So neutral lysine, basically it will just be NH2. So there are two protons in the lysine side chain. And so two peaks are appearing. You can see these are all lysine, lysine, lysine. So two peaks are appearing because there are two hydrogens in the lysine side chain. So we will look more into detail in the toxi and cozy in the next slide. One thing to remember is that sometimes in toxi, if you remember, toxi basically shows correlation between all the protons. So if you are looking at lysine, you will see alpha correlated with beta, correlated with gamma, delta, epsilon, so on. So through the entire chain, there will be correlations. But sometimes if the mixing time of the experiment is short, the polarization will not be able to be carried through the entire chain. So sometimes the protons at the end of the chain, like the delta or epsilon, may not appear in the spectra. So that is one thing that you have to ensure, that the mixing time is long enough. So now we will look at more details of this 2D NMR spectrum. So cozy spectra, as you know, cozy spectra basically only shows coupling between adjacent spins. So basically 3J coupling. Cozy spectra is 3J coupling, where you have three bonds. So separating these two hydrogens, you have 1, 2, and 3 bonds. So cozy spectra will only show correlations between adjacent or vicinal hydrogens. So you have different regions of the cozy spectra. You can have, so this here, this F region is basically correlation between aromatic protons. If you have an aromatic ring, so correlation between these two protons or these two protons is found in this F region. In the G and H region, it is basically between NH C alpha. That is the backbone amide NH and the C alpha proton. Then as you go more from the left to the right, you will see different or adjacent correlations. So alpha H to beta H, then beta H to gamma H, then gamma H to delta H, so on. You will see different correlations as you go from the left to the right. Now, one thing about the cozy spectra is that if you look at this region, which is this G and H region, so you at the bottom, you have the range from around 9 to 6.5 or 7. And here you have the range around, say, 3 to 5. Or you can even extend it 1 to 5. which is shown here. So now we are looking at the G and H region. That is the region where you see the NHC alpha correlations. So number of peaks in this region is equal to number of amino acids, because each amino acid will only have one backbone NH and one backbone C alpha. That is, in glycine you can have two, but NH will always be one. So this kind of if you look at this region of the COSY spectra, so basically this shown here is an expansion of this part. Number of peaks that you see in this region is the number of amino acids in your peptide chain. So you have 5 peaks here. That means the peptide chain has 5 amino acids. But if you look at the toxi in the same region, number of spins is much higher, because toxi will not only show up to alpha. Suppose we are looking at toxi, then from alpha to beta also, alpha to beta you will see, alpha to gamma you will see, alpha to delta. So toxi will show all the correlations. So the number of peaks in toxi is much higher. So looking at toxi, basically what we do, we look again at the vertical lines. So this peak in toxi is the same as COSY. So this is a NH to C alpha correlation. Then if you go in the vertical line, you will see the C alpha C beta correlation. That means this amino acid only has C alpha and C beta because there are 2 peaks. So it can be something like, we can see which amino acids only have C alpha C beta. So it can be something like alanine, which only has C alpha and C beta. Or it can be something like serine, which has C alpha C beta. Important thing to remember is that when you have polar hydrogens like OH or NH, they are often not visible in NMR because they will exchange with the H2 or D2 or whatever solvent there is. And we know that exchangeable protons are often not seen. That is why in DNA, we were often not able to see the imino or amino protons. Similarly, we can look at the other peaks. So if we look at this vertical line here, this peak is the same as we saw here. So this is your NHC alpha peak. Then you can have beta, gamma, delta. So this amino acid is something which has up to delta carbon. So up to delta carbon means which amino acid can it be? It can, we look for an amino acid where you can see up to delta correlations. So it can be asparagine. You can see up to delta hydrogen. Or it can be, it cannot be aspartic acid because here the delta hydrogen is exchangeable. It will exchange and so you will not see the delta hydrogen in case of asparagine. Or it can be, it can even be leucine. In fact, leucine is the most probable because asparagine, you have gamma carbon, but gamma hydrogen is not there. So toxi is hydrogen-hydrogen correlation. So if we see a correlation up to delta hydrogen, it is most probably something like leucine, where you have H alpha, H beta, H gamma, and H delta. So like that, looking at the toxi spectra, you can predict that which is the amino acid that is present. Then these are the expansions of these regions, which are much more crowded. So again, you can use the similar ideas here, that you can, we will look into other toxi spectra, like for each amino acid. But so right now it appears very crowded and difficult to identify. But in the subsequent slide, we will look at the different toxi spectra of different amino acids. So each amino acid basically has a very characteristic COSY or toxi spectra. And using that, you can identify, looking at the toxi, that what is this amino acid. So now we will look into that one by one. So first we will look at the aromatic amino acids: histidine, phenylalanine, tyrosine, and tryptophan. So we will look only at the aromatic ring region. So we saw that the aromatic ring region is somewhere around here, 8 to 6.5 or so on. So we are only looking at that region. So we are ignoring the other backbone amide or this CH2. So we can see that in the aromatic ring, histidine basically has two protons. So we label this position as 1. Then this hydrogen can be written as H2. This will be 3 and this will be H4. So histidine ring only has two protons, 2 and 4. This hydrogen attached to NH, I have already discussed that hydrogens attached to residues like N or O are exchangeable with the D2O or H2O solvent, and so they will not appear in NMR. So the diagonal peak, the peaks in the diagonals are basically the ones which are the hydrogens present. In histidine, it will be 2H, which is this one, and 4H, which is this one. So there will be a toxic cross peak between 2 and 4H. You can see between 2 and 4H, there is one cross peak. So if we look at the toxic spectra and it appears like this, we can say that the histidine is present. In case of phenylalanine, it's more complex. So we'll first look at tyrosine because the number of protons in tyrosine is less. So in tyrosine, you have two protons ortho to the OH group. We have labeled that as A and A prime, and two other protons which are meta to the OH group labeled as X and X prime. Again, we are only looking at the aromatic ring. We don't consider the OH proton because it is exchanging with the solvent. So in the phenylalanine aromatic ring region, we will see four protons, A prime, and X prime. So it is written here as A prime, X prime. But the chemical shift of A and A prime will be same because they are in similar chemical environment. They are just adjacent to the OH. So there will be one peak here, and imagine that. So A prime will be more downfield shifted because they are adjacent to the OH. So I will call this peak as A prime. And this peak as XX prime. Again, XX prime will appear at same chemical shift because their chemical environment is similar. So it is appearing here. Then between A and X, you will see one toxic cross peak, and between A prime, X prime, you will see another toxic cross. Both these will appear at the same position. So this kind of pattern of the toxic spectra can be assigned to that of tyrosine. In phenylalanine, instead of this OH, you will have an H group. So A prime, X prime remains same, but you have an extra third kind of proton, which we are calling as M. So the system is called as A prime, X prime, M. There are three peaks here as so again we have this peak is the A prime peak. This peak is M, and this peak is XX prime. Now, if we are looking at just the COSY spectra, COSY means only adjacent. So if it is adjacent, you will see between A, M, A prime, M. So that peak is this circle shown here. Then you can have between A, X, A prime, X. That is this circle shown here between A and X. But what about this peak, which is here? This peak is between M and X prime. Now, M and X prime are too far away to interact by COSY. But if it is a toxic spectra, you will see this peak also, which is shown by the cross here, because toxi shows all the correlations. Then you have tryptophan. In tryptophan, this NH is again exchangeable, so it will not be seen. In this six-membered aromatic ring, you have four kinds of protons and one proton in the five-membered ring. So total you have 5 different kinds of protons. So you can see 5 peaks in the diagonal. Labeling is basically, this is labeled as 2. So this is H2 here. This 2 is not coupling with any other hydrogen in the aromatic ring system. So you can see 2 has no cross peaks. The other cross hydrogens, so there are 4 other hydrogens, 2, 3, 4, 5. We can label this as 2, 3, 4. Yeah, we can give different numbers. So these have been labeled according to that particular system. Basically, you can see there are 4 hydrogens here and 4 other peaks in the diagonal. The cozy cross peaks will only be the adjacent cross peaks. So 1, 2, and 3. You will have 3 cozy cross peaks, which are shown by the 3 white circles here. And if you are looking at toxy, then toxy will have other extended. So between this and this, toxy will be there. Between this and this. So toxy will have other peaks, which are shown here. So tryptophan is the most complex spectra of all the aromatic amino acids. Then you have uh, another kind of amino acid called the ABX system or type J. So in this amino acid, what happens? You just have alpha and beta protons. If we look at serine, so in all the examples shown here, C alpha, H alpha is basically not shown. So you just imagine one C alpha, H alpha here. So serine has C alpha, H alpha, then C beta, H beta. OH is exchangeable, so it is not seen. Then aspartic acid, again C alpha, H alpha, C beta, H beta. And this H is again attached to the carboxylic acid group, so it is not seen. Similarly, for asparagine, cysteine, they also only have C alpha and H alpha if we ignore the polar protons which are exchanging. Then the aromatic amino acids, so far we have looked only at the aromatic chain. So now we will look at the portion excluding the aromatic chain. So up to here or here. So we are just excluding the aromatic chain. So in all the aromatic amino acids also, you will only have C alpha and C beta. That is H alpha, H beta. So in these 8 amino acids, serine, aspartic acid, asparagine, cysteine, tryptophan, phenylalanine, tyrosine, and histidine. If you are only looking at the aliphatic region, that is, we are not looking at the aromatic ring regions, you will see this kind of pattern where you have H alpha and 2 H betas. And H alpha is correlating to H beta 1, it is correlating to H beta 2, and the 2 H betas are correlating between themselves, which is the geminal coupling. And so this kind of pattern will be seen. In this case, toxy and cozy will be the same because you only have adjacent H alpha, H beta protons. So these are called ABX system because you have 3 kinds of spins, H alpha, H beta 1 and H beta 2. And they are also called type J because, as I said, it is coupling only between adjacent protons. Here you have the aliphatic couplings. So you can have, so these 8 fall within the same category. These 3 are different categories. Valine basically will have H alpha, H beta, then H gamma 1 and gamma 2 because the 2 methyl groups will become unequivalent. You can see along the diagonal you have alpha, H beta, and then the 2 methyl groups in the valine are not equivalent. So you have gamma 1 and gamma 2. So again, as expected, in cozy, you will see only the adjacent. So cozy is basically these white circles and toxy are these small circles shown here. So cozy will only show adjacent correlations, alpha to beta, beta to gamma 1, beta to gamma 2. And toxi will show longer correlations like alpha to gamma and so on. Then you can have leucine, where after gamma, you have delta 1 and delta 2. So the two methyl groups at the delta position are not equivalent and they have been written as delta 1 and delta 2. So along the diagonal, you will have alpha, beta 1, beta 2. The two betas are also not equivalent. Then only one gamma, because there's only one gamma hydrogen. Then delta 1, delta 2 because of the two delta CH3 groups. Then, as expected in cozy, you will only see adjacent correlations, and in toxi, all the correlations will be seen. And in isoleucine, you will have H alpha, H beta. There is only one H beta. There are two different H gammas. One is a CH2 group, one is a CH3 group. So you have gamma 1. So there are actually three different H gammas. One is this CH3 gamma, one is CH gamma, and another is another CH gamma. So the two CH are also not equivalent. So like you have beta 2 and beta 3, the two beta hydrogens are not equivalent. Similarly, the two gamma hydrogens are not equivalent, gamma and another gamma. So you have three kinds of gamma hydrogens, gamma hydrogen 1, gamma hydrogen 2, and gamma CH3. You can see here the three kinds of gamma. And then you have one delta CH3. So isoleucine also has, as you expect. So in all these, you have alpha as the most downfield shifted. And because it is nearest to the, so if you imagine alpha is here, alpha is attached to your NH group, it is attached to COH group, which are electron withdrawing groups. So alpha is the most downfield shifted because of the electron withdrawing amino and carboxyl groups. And as you go down the chain, they are more upfield shifted. You have beta, gamma, then delta. So further you go away from the electron withdrawing amino and carboxyl groups, the shift becomes more upfield. These are just the aliphatic residues where you do not have any kind of polar atoms. There is another kind of spin system, which are basically long side chains. And long side chains having polar atoms. So leucine is also a long side chain, but it is long side chain having only CH atoms. And these 6 amino acids, lysine, arginine, methionine, glutamine, glutamic acid, and proline, they fall under another category called as long side chain, but with polar atoms. And they are often called as type U. So we will look at how these appear. So first we will look at glutamine, glutamic acid, and methionine. Here one thing to note is that in these amino acids, we always had the chemical shift go from alpha to beta to gamma, then delta. So as we move from alpha to delta, it went more upfield. But in this case, you can see that alpha is most downfield, but gamma is more downfield compared to beta. Why is that? Is because in gamma, another electron withdrawing group is attached. So you can see these are the gamma hydrogen in glutamine, and the electron withdrawing CONH2 group is attached. These are the gamma hydrogen in glutamic acid, and the electron withdrawing COOH group is attached. Similarly, methionine, these are the gamma hydrogen, and electron withdrawing sulfur is attached. So because of this electron withdrawing effect of these groups, the gamma hydrogen is getting more downfield shifted compared to the beta hydrogen. So if you look at the diagonal, you have the alpha, then you have gamma, and beta is most upfield. But Cozy will always be between adjacent peaks. So the white circles are the cozy. So alpha and beta will show cozy. Then gamma and beta will show cozy. And between two beta also there is one cozy peak, and between the two gamma also there is a cozy peak. So this is one kind of system. Another is where you have proline and arginine. Now in proline and arginine, you can see alpha, then delta, then beta, then gamma, if you look along the diagonal. So delta is quite downfield shifted. This is because proline, if you look at edge delta, it is attached to the nitrogen. Then arginine also, if you look at edge delta, it is attached to the nitrogen. So because of the electron withdrawing effect of nitrogen, delta is getting downfield shifted and coming close to alpha. And lysine also, if you look, you have alpha, beta, gamma, delta, but epsilon is getting so much downfield shifted. This is because in lysine, it is the epsilon hydrogen, epsilon carbon to which the nitrogen is attached. So depending on where the electron withdrawing group nitrogen is attached, that the hydrogen attached to that carbon will get downfield shifted. And accordingly, the patterns will change. So these six amino acids where you have long side chains, but you also have a polar residue attached to the side chains, are called as long side chain or type U spin systems. So now we have looked at Toxi and Cozy spectra, and we have used Toxi and Cozy spectra in order to identify the spin system. That is, to identify which amino acids are present. Then but we don't know the order of these amino acids, because we have seen before also Toxi and Cozy, they occur within the same amino acid. In order to understand the sequence of the amino acids, nosy is used. And nosy, we know that if two protons are within 5 angstrom distance of each other in space, we will see nosy interactions. So in nosy spectra, three kind of peaks can happen. One is the near neighbor interactions, meaning to the amino acid just after it or just before it. So if you look at the thick arrows here, so this amide hydrogen to the next amide hydrogen, I to I plus 1, between the amide hydrogens, this is called as DNN. Then again you can have from I plus 1 to I plus 2. So from I plus 1, you can go to the previous residue I, or you can go to the next residue I plus 2. This is called near neighbor interactions because it is only between either the amino acid directly after or the one directly before. Then another kind is the alpha N. So alpha N is happening again between adjacent amino acid residues. So this is also a near neighbor interaction. Then you can also have beta N. So beta N is between, so this was alpha hydrogen to NH. So this is alpha N. Beta N is from beta hydrogen to NH. So this is beta N. So these are near neighbor interactions where it is only happening with the amino acid directly after or directly before it, or the sequential amino acid. Then you can have short range interactions, which are interactions which are 3 to 4 residues apart. So if you look at the dotted lines here, the alpha of I is reacting with the interacting with the NH of I plus 4. So this interaction is called D alpha N I plus 4, because alpha of I and N of I plus 4. So this is called a short range interaction, which are interactions between residues which are 3 to 4 residues apart. So the near neighbor interaction will help you determine the sequence of amino acids in the primary structure. The short range interactions will help to determine what is the secondary structure. So as you can see here, different secondary structures. So we look at alpha helix. I'll just discard all this. If we look at alpha helix has very distinct distances. So we will first look at this d alpha n. D alpha n is a near neighbor interaction, and using this we can find out the order of the amino acids. Then we will look at d alpha n I plus 2. So that is this interaction d alpha ch alpha of I with nh of I plus 2. This is a short range interaction. This interaction, we will get a nosy peak from this interaction, and from the intensity of the nosy peak, we will get a distance. If that distance is around 4.4 angstrom, then a alpha helix is formed. Similarly, from each of the nosy peaks, we can get a certain distance or distance range. And from that distance, so each secondary structure has a very characteristic distance or distance range. So from the nosy, we will derive these distances, and whichever secondary structure the distances match with, that is the secondary structure our protein is adopting. So using nosy, we can not only find out the sequence of amino acids in the protein, but we can also find out what is the secondary structure the protein is adopting, whether it's an alpha helix, a 310 helix, antiparallel or parallel beta sheet, or whether it is a turn. Turns can also be of different types. Then we can have very long range interactions, which are interactions between residues that are 10 to 15 amino acids apart. That is not shown here, but these normally help in understanding the tertiary structure of a protein. So suppose you have an alpha helix here, then a protein which has a beta sheet, and so on. Now, if there is an interaction between, suppose, this region and this region of the, so there is this kind of interaction. So the residues are far apart from each other, but because of the folding of the protein, they are coming close to each other, and we can see a nosy interaction. So this kind of interaction is a long range interaction where the residues can be 10 or 15 amino acids apart, but due to folding of the protein in the tertiary structure of the protein, they are coming close to each other. So the long range interactions can help to determine the tertiary structure of the protein. So we have already seen this before in COSY. COSY, if we are looking at the NHC alpha cross peaks, and we have said that number of cross peaks in this region will be number of amino acids. So if we have 5 cross peaks in the NHC alpha region, means that there are 5 amino acids. Now, the nosy of this, basically, nosy, we have just seen that this NHC alpha, or which we can call alpha N. Alpha N is a sequential interaction which happens to the from I to I minus 1. So we have seen here, suppose we are looking at I plus this I residue. So alpha N is the one shown here between alpha of I and N of I plus 1. Then you can again have alpha of I plus 1, N of I plus 2, and so on. So you can also have between alpha and N of the same residue. So alpha of I plus 1 and N of I plus 1. So alpha N nosy cross peaks can occur. You can have self peak, that is H alpha I to NHI, and you can have sequential peaks H alpha I to, so from NHI, you can have self peak to H alpha I, and you can have 2 sequential peaks from to H alpha I minus 1 and H beta or I minus 1 also. So we already know that from nosy, we can do kind of a walk through the nosy spectra. So we will look at this. So suppose we are starting from here. One second. Yeah. So in the nosy peak, what we can do, suppose we are starting, so each nosy will have a self peak that is within the same residue, and it will have different cross peaks with the residue before it or after it. We have already seen this in DNA, how we can walk through the nosy spectra in order to find out the sequence of nucleotides in our DNA. Similarly, we can walk through the nosy peaks of the amino acid chain to find the sequence of the polypeptide. So right now, here, suppose we are starting from here. This amino acid is a terminal amino acid, and so it will have, from here you go to the sequential peak. So this amino acid has a sequential peak here. Now, if you go in the vertical direction, it will be the self peak. From the self peak, again, you go to different sequential peaks. So like that, you can connect the different peaks of the nosy spectra from self peak to sequential peak to get the order of amino acids in the polypeptide. Similarly, this is the case of alpha helix. Similarly, in case of beta sheets, you will get a self peak between, so here in beta sheet, what are the peaks you will see? You will see two peaks from NHI to H alpha I. So you can see this NHI to H alpha I shown by this arrow, and NHI to H alpha I minus 1. To NHI to H alpha I minus 1, again NHI to H alpha I minus 1, so on. So in the beta sheet, suppose I start from here. So if this is the self peak, then this is the sequential peak. So suppose this is I. This is the sequential peak with I minus 1. This is again, when we go in the horizontal direction, this is the self peak of I minus 1. Then as we go in the vertical direction, we get I minus 2. Then we can again go in the horizontal direction to get I minus 2. So like this, we will move through the polypeptide in order to get the different peaks of each adjacent residue, and we will understand the polypeptide sequence. Just give me 1 second. Yeah, sorry, I think the voice was muted. So basically, what I was saying is that in a 7 residue helix, you will find 3 I to I+4 interactions. You will 1 to 5, 2 to 6, and 3 to 7. That is what's shown here. Then if it's I+3 interaction, you will find 4 I to I+3 interactions. 1 to 4, 2 to 5, 3 to 6, and 4 to 7. Similarly, you can find out different near neighbor interactions, and these interactions will help you understand the secondary structure of the peptide. So you were initially finding using the immediate neighbor interactions or the sequential interactions, you found out the sequence of amino acids in the peptide using this kind of a walk through the nosy. Then using the near neighbor interactions, you can find out which secondary structure it is adopting. Finally, the use of the long range interactions. So long range interactions we discussed before also are between 10 to 15 residues apart, and they show us that what is the tertiary structure of the folded protein. So suppose you have a polypeptide chain here, and there are 4 residues in this chain shown by A, B, C, and D. Now, what are these residues? That has already been identified through sequential connectivities. Now, suppose this chain is folding in this manner so that the terminal residues are coming close to each other, like A and D. So A and D will show a long range nosy. And if you see that nosy, you can predict that this is how the protein has folded. Similarly, if A and B show a nosy, we can predict this is how the protein has folded. Or if there is a BC nosy, then this is how the protein has folded. So depending on the folding, the long range nosies will be different. And so nosy is often called the fingerprint of the structure of the molecule. So this we have seen in the previous class that from the nosy, we will get, from the intensity of the nosy, we will get a range of distance. We will get an upper limit and a lower limit of distance between the protons. So that is basically a distance range. And using this distance range, there is an energy function. In the energy function, there is a constant term EF that is made up of the intrinsic energies. That is the bond distance, bond angles, and other torsional angles, so that there are set in such a way that there are no steric clashes. And you have a NOE dependent energy where this formula is used. Basically, you take the deviation of the interproton distance from the lower limit and you square it, multiply with a force constant, and you sum it over all the proton pairs which are interacting. And the same is done with the upper limit distance. The deviation of the distance from the upper limit is taken, it is squared, multiplied with a force constant, and summed up over all the proton pairs that are interacting. Now, this ENOE, basically, we have to keep optimizing the structure till the ENOE is 0. And when the ENOE will be 0 is when all the NOE distance constraints are satisfied. So, there are certain distance constraint or distance restraints which we are deriving from NOE. If the structure that is optimized has all the distance which are not violating any of these distance restraint, then this ENOE will be 0. And so we have to continue optimizing the structure till all the violations of the distance restraints are removed and the ENOE becomes 0. That will be the structure that satisfies the NMR. So like we have seen in the optimization of DNA as well, there was this Tandy algorithm, that is the torsion angle based distance geometry algorithm. So we start from many different initial structures. You can see here 6. This is 1, 2, 3, 4, 5, and 6. 6 different initial structures are there, and we choose different initial structures because we don't want the calculation to be biased by the choice of the initial structure. So each initial structure is optimized after putting the distance constraints, torsion constraints, etc. that we have obtained from NMR. So the constraints in all the structures are the same, and then we allow it to optimize till all the constraints are satisfied. How will we know all the constraints are satisfied? This ENOE will be 0. So we continue the optimization till all the constraints are satisfied and we get a final structure. We can see that the final structure is the same no matter what initial structure we are starting from. So we can say this, that the set of constraints that we have used are able to give us a unique structure. So just one second. So then we can say that the set of constraints is also correct, because no matter what initial structure we are starting from, we are reaching the same final structure. Now, we have seen this before also, that when the two-dimensional spectra gets too crowded, then we extend it to 3D or if required 4D NMR. And the basis of this is that suppose we have a 2D NMR, look at the lower plane here, where we have H on both the axes. Now, in both the axes, so this kind of HH nosy is very crowded, as you can see here. Now, if we add a third axis consisting of 15N, so this example shown here, it is basically pulling the nitrogen chemical shifts up. So each plane is corresponding to only one amino acid. Because the N15 chemical shift is different for the different amino acids, so each plane will have one amino acid. And within that plane, you can have the alpha, beta, gamma hydrogens of that particular amino acid. So in this system, if you have 3 amino acids, the HH spectra can be very crowded, because suppose in the same plane you have alpha, beta, gamma of one amino acid, alpha, beta, gamma of another amino acid, and so on. But if you introduce a third axis, they will separate out, the different amino acids will separate out in different planes, and it is easier to resolve this kind of 3D spectra. So we have already studied the different kinds of 3D spectra in the previous classes. So we won't go into the details of that right now. This is just a kind of reminder that this kind of triple resonance experiments that we have studied, basically they are used to assign the structures of long proteins consisting of more than 150 amino acids. So we will look at the most common experiment, that is the HNCA experiment. So we start from the amide hydrogen of the I residue, HNI. The magnetization is transferred to the nitrogen of the I residue, Ni. And here you have the first T1. So T1 is basically an evolving time period. And based on T1, we will get our frequency axis. So the first frequency axis, or F1, corresponds to N15. So F1 shown here is N15. Then from the N of the I residue, the magnetization is transferred to I minus 1 and I. So magnetization is transferred to C alpha. These are both C alpha. Magnetization is transferred to C alpha of the I residue as well as C alpha of I minus 1 residue. You can see here C alpha I minus 1. Then here is where the second evolving time period is introduced, T2. So F2 is your C alpha. So we saw F1 was 15N, F2 is your 13C. And now the magnetization comes back. So from C alpha, it again comes back to Ni, and from Ni, it again comes back to this initial amide hydrogen where the T3 is recorded. So your F3 is basically 1H of the amide hydrogen. So you have three different dimensions. Now, if you take different planes, so here we have taken one plane shown in the dotted lines here. So this plane corresponds to a specific value of F1, but F2 and F3 are changing. So in the plane, in the x-axis, we have the Hn, that is the amide hydrogen chemical shift. And in the y-axis, we have the 13C chemical shift. So suppose we have taken one plane corresponding to a specific value of F1, that is the N15 chemical shift of I-1. So in that plane, we will see two cross peaks. One is the self peak, so N15 of I-1 to the NH of I-1 and N15 of I-1 to the so so N15 you have fixed the N15. In that plane, you will see two peaks. One is the self peak, which is shown in dark red here, and one is the sequential peak, which is shown in this hollow circle here. So self peak of I-1 is with I-1 itself. Sequential peak is with the previous residue, so it is with I-2. Then if you take another plane like this corresponding to N15 shift of ith residue, again here you will see two peaks. So here again you will see, suppose one self peak here, it is I, so I will be the self peak, and you will see one peak with the previous residue I-1. So how this will help is that you can walk along the polypeptide chain. You can take different planes in this three-dimensional spectra. So here, different planes are laid out. Each plane corresponds to a specific value of F1. So the first one is N15 shift of I. Second is N15 shift of I-1. N15 shift of I-2 and I-3. So in each peak, you are getting a self peak, which is shown by the dark red circle, and a sequential peak to the previous residue shown by this empty circle. So in the I plane, this peak corresponds to I, this peak corresponds to I-1. So I-1 cross peak will, if you draw a line from that, that will lead you to the self peak of I-1. This is I-1, and now I-1 will show a cross peak with I-2. Then you can go to the next plane. So this is I-2. I-2 will show a sequential peak with I-3. So like that, you can find out the sequence of the amino acids. And how can you distinguish that which is the self peak and which is the sequential peak? Basically, the HNCOCA experiment helps in doing that, because in HNCOCA, it is like HNCA, but in HNCA, from the nitrogen, it is directly getting transferred to C alpha on both sides. In HNCOCA, the carbonyl group is used as a kind of intermediate to help transfer the magnetization. And because the carbonyl group is only on one side, the C alpha of only the I-1 is detected. So if you look at the HNCOCA thing, there will be no self peak. So here there was transfer from N to C alpha I, which was the self peak. Here the self peak is removed. You will only see correlation with C alpha I-1. So there is only cross peak. So comparing the chemical shift of these two, you can find out which is the self peak and which is the sequential peak. These are examples of 3D spectra. We have already shown this before. Basically, if you draw projections onto any plane, you will get the corresponding 2D spectra. So suppose this plane, the bottom plane, is a 15N versus 13C spectra. Then this plane on this side is a 1H versus 15N spectra. So from 3D spectra, you can get the corresponding 2D spectra by projections on that plane. Then this is basically shown that as the NMR techniques are getting more and more advanced, so from 2D, different kinds of 3D techniques are now in use, and even 4D techniques are now in use. So as the different NMR techniques and pulse programs are getting more and more advanced, the number of protein structures deposited, that is, for a protein, basically you find the NMR and then you derive the structure. So number of protein structures derived and deposited have rapidly increased with increase in refinement of the NMR techniques. And we have also looked into the HNN experiment before. Basically, there are some proteins called intrinsically disordered proteins where the 13C or 1H, so basically in HNCA, we were looking at 1H, 13C and 15N. But if the 1H and 13C are overlapping a lot, then it is very difficult to use HNCA. And this happens in the case of intrinsically disordered proteins. The 1H and 13C overlap a lot. So instead of that, the HNN experiment is used. We have already studied this before. In HNN, basically, you start by exciting the amide hydrogen of the ith residue. From there, the magnetization is transferred to the nitrogen of the ith residue, and this forms your first frequency axis. So T1 is 15N. Then from I, the magnetization can transfer to C alpha of I and I minus 1. So far, it is same as HNCA. This is where the difference comes into play. From C alpha in HNCA, from C alpha, you would have gone back to the same nitrogen. But in HNN, you go to the nitrogen. So from C alpha I, you can go to nitrogen of I or nitrogen of I plus 1. And C alpha I minus 1, you can go to nitrogen of I minus 1 or nitrogen of I. This forms the second frequency axis. So T2 is again 15N. And from the corresponding nitrogens, you go to the corresponding amide hydrogens, which form the T3. So T3 is your amide hydrogen. So HNN is basically a more defined form of HNCA. So you can see here, suppose we are starting from this H. This H, we will come to this N. From this N, we will go to C alpha I and C alpha I minus 1. From this, we will go to, from C alpha I minus 1, we can go to Ni minus 1, or we can go back to Ni. And from C alpha I, we can go to the next N, Ni plus 1, or we can again go back to Ni. So at the end of the day, we are detecting NH, I minus 1, I and I plus 1. We are also detecting Ni minus 1, Ni and Ni plus 1. So HNN experiment is called a triple filter experiment because you are able to identify 3 consecutive residues, I plus 1, I and I minus 1. So because you can simultaneously identify 3 consecutive residues, it is called a triple resonance experiment. So now we'll just quickly go through some the assignment of week 6. And so we have covered the summary of all the videos of week 7. So now we'll go into week 6, the assignment. So one question was that BDNA is what? Left-handed or right-handed, with a helix pitch of how much, and sugar puckering is what? So we know that the bDNA, which is the most common form of DNA, is a right-handed helix. You can see the helix shown here is right-handed. So automatically options A and D cancel out. Then the pitch of the helix. The pitch of the helix is the rise in helix per turn. So this is one turn of the helix. The rise in helix per turn is 34 angstrom or 3.4 nanometer. So this cuts out, and your correct answer is option B. We'll just look at the sugar puckering also. Sugar puckering, basically there are two main kinds of sugar puckering. One in which the 3' atom is above the plane, 2' is below the plane. This is called 3' endo. Endo means above the plane. This belongs to A DNA, but here we are talking about B DNA. In B DNA, 3' is below the plane and 2' is above the plane. So this will be called 2' endo because 2' above the plane. So in B DNA, the sugar puckering is C2' endo, which is here it's C2 endo. So B is the correct answer. Then you have a question asking that nitrogen of 5-membered ring of purine is hydrogen bonded to pyrimidine in what kind of base pairing? We had looked in the previous class that there are three main kinds of base pairing. One is the common Watson-Crick, where A pairs with T or U, depending on whether it's DNA or RNA, and G pairs with C. So in AT or AU, there will be 2 hydrogen bonds, and in GC, there will be 3 hydrogen bonds. In this Watson-Crick base pairing, you can see that in A or G, that is the purine, it is only the 6-membered ring that is participating in hydrogen bonding. The second kind of hydrogen bonding is Hooke's-Steen hydrogen bonding, where the pair is similar. You have A pairing with T or U and G pairing with C. But in case of Watson-Crick, the purine, only the 6-membered ring was contributing to the hydrogen bonding. In Hooke's-Steen, you have one nitrogen of the 5-membered ring and one hydrogen of the 6-membered ring. So both the 6-membered ring and the 5-membered ring are participating in the hydrogen bonding in Hooke's-Steen base pairing. And the third kind of base pairing is called Wobble base pairing, where the pairing itself is different. So G does not pair with C, it pairs with U. So when the base pairing is different from the expected Watson-Crick, that is called Wobble base pair. And in these three different kinds of base pairing, Watson-Crick, Hooke's-Steen or Wobble, you can have, for each you can have a reverse pairing. So reverse is basically, if you imagine this dotted line and you flip the second residue 180 degree. So imagine a dotted line here and you flip the second residue 180 degree about this line. This NR is pointing down, NR will now point up. So the pairing remains same. It is just that the second residue is flipped through 180 degree. That is called a reverse. So each Watson-Crick can have reverse Watson-Crick, Hooke's-Steen can have reverse Hooke's-Steen, and Wobble can have reverse Wobble. The question is asking, nitrogen of 5-membered ring of purine is hydrogen bonded to pyrimidine. This happens only in case of Hooke's-Steen. So 5-membered ring of purine participates in base pairing only in case of Hooke's-Steen. Because in both Watson-Crick and Wobble, it is only the 6-membered ring of purine that is participating in base pairing. So the correct answer is C, that is Hooke's-Steen base pairing. Then you have another question. If you are getting peaks in the range 0 to 1.5 ppm, these peaks belong to what? So if you remember the previous class, 0 to 1.5 ppm is a very unique range. And in that range, only methyl groups can appear. this range, which is less than 2 ppm, only CH3 groups can appear. And in all the nitrogenous bases, only thymine has a CH3 group. So if you are getting peaks below 2 ppm, that means they must belong to thymine because it is the only nitrogenous base that has a CH3 group. So the correct answer is C, that is thymidine from DNA. Then you have a question asking that for the structure calculation of nucleic acid, which of the following information is required? So if you remember the formula for structure calculation, the first thing you need to do is derive distance constraints from the nosy intensity. You know, nosy intensity is directly proportional to 1 by r to the power 6, where r we can write as interproton distance, ij. So from nosy intensity, we need to find the distance constraints. So nosy information is important. Then we have an energy function. So we need to have an which has two components. The ENOE component depends on the nosy, but it is this component that we will look at now. So this component is the intrinsic energy of the compound. So the energy, just 1 second. Right. So as I was saying, we will now look at the EF term. The EF term consists of intrinsic energies based on the bond distances, bond angles, and so on. So energetic information is also needed. And we also need the sugar geometry information, because depending on the sugar geometry, that is the torsional angles, puckering, and so on, the EF term will vary. So sugar geometry and energetic information is necessary for the EF term. So you need all three, A, B, and C, for structure calculation. And so the correct answer is all of the above, that is D. Now, this is a question asking that how many coupling constants are present in sugar moiety of nucleotides. If you remember the previous class, so here we have a sugar molecule being shown. What are of DNA? What are the couplings that can happen? You can have 1' to 2' coupling, 1' to 2'. You can have 1' to 2 double prime, so the other 2'. Then you can have 2' to 3'. You can have 2 double prime to 3'. And finally, you can have 3' to 4'. So these are the 5 coupling constants that can happen in case of DNA. So if it is RNA, then instead of 2 primes, you will have one OH. So then the numbers will be different. But right now we are looking at DNA. So there are 5 coupling constants. So the correct answer, that how many coupling constants are present, is 5. That is option A. Now we have seen in the previous class that how these 5 coupling constants can vary according to the pseudo rotation angle. So we won't go into all that right now. It is enough to remember, if you just remember the structure of the sugar moiety, you will know that there are 5 coupling constants. Because when people talk about coupling constants, if nothing is mentioned, we normally consider only the 3J coupling. So the adjacent coupling, and the 3J coupling constant I just showed, there are 5. Then you have a question asking, which experiment is used to determine sugar geometry fine structure? So if you remember the previous class, first of all, the sugar geometry, the cross peak, we can see by COSY. So we just saw, so C2 prime endo is the orientation where the 2 prime atom is above the plane. So that is your C2 prime endo orientation. And in the previous um, slide, we saw that in C2 prime endo, you will basically have 3 coupling constants. H1 to 1 prime to 2 prime, 1 prime to 2 double prime, and 2 double prime to 3 prime. So there are 3 coupling constants in the sugar ring of C2 prime endo. These two are too small. You can see this is around 0 Hertz, this is 1 to 2 Hertz. This is too small and they will not appear in the 2D spectra. So there are 3 coupling constants in C2 prime endo. So from the 3 coupling constants, you will get 3 peaks in COSY. 1 prime interacting with 2 prime, 1 prime interacting with 2 double prime, and 2 prime interacting with 3 prime. So 3 peaks in COSY. From the COSY itself, you can find out the fine structure of the COSY. We have discussed this in detail in the previous class. I'm not going to go into that much detail right now. It is enough to remember that using the COSY, we can find out the sugar. So if we can see 3 peaks in the COSY, we will know it is the C2 prime endo geometry. So what did we use to determine the sugar geometry? We used the COSY experiment. So the correct answer is option B. Similarly, if the sugar geometry was C3 prime endo, we would have seen so in C3 prime endo, if you can see, one coupling constant is 0. The other 4 coupling constants are greater than 6 Hertz. So 1 prime, 2 prime, you will not see. So imagine this was, if this was C3 prime endo, then you will not see 1 prime, 2 prime. So we will not see 1 prime. So this peak we will not see, but all other peaks you will see. 1 prime, 2 double prime, 3 prime with both, and then 3 prime, 4 prime also you will see. So you will see 4 peaks. So if you see 4 peaks in COSY, that means it is the C3 prime endo. So using COSY, you can find out the sugar geometry fine structure. Then another question was that COSY 45 gives a better fine structure due to what? So if you remember, COSY 45 is the experiment. So after the first 90-degree pulse, there is an evolution time, and then there is a second pulse. If the second pulse is 90, it is a normal COSY experiment. But if it is 45 or smaller, it is called COSY 45 or E-COSY. Basically, in this, what happens? So this is the fine structure of a normal COSY experiment. In COSY 45, what happens? All these cross peaks that you have, the intensity is altered. So normally in this COSY fine structure, which is shown in the previous slide, you can see there are plus, minus, plus, minus. So if there are opposite signs, they can cancel out. And the number of peaks in the fine structure, suppose in C2 prime endo, 16 cross peaks are expected in the fine structure. But we were observing only like 4 cross peaks because all the opposite signs are getting cancelled out. But in E-COSY, what happens? The intensity in the cross peak is altered so that the cancellations are getting reduced and we are able to see the fine structure better. So the fine structure is getting resolved more because the intensity in the cross peaks are getting altered and the cancellations of opposite signs are getting reduced. This gives us a better fine structure. This helps us to better derive the coupling constants from the fine structure. So the correct answer is B, that in COSY 45, the intensity in the cross peak is getting altered. Then you have a question asking the lowest energy for epsilon torsion angle is what? So epsilon torsion angle is basically the torsional angle between C3 prime and O3 prime. And this is a very important constraint. As you are rotating along this bond C3 prime O3 prime, you can see the energy versus the epsilon angle reaches a minima around 270 degree. If you move slightly away from 270 degree, the energy is increasing very sharply. So the epsilon is constrained to very small values around 270 degree only. So in the entire 360-degree range of angles, only a very short range of angles is allowed for epsilon, because away from the angle, the energy is very unfavorable. So epsilon is actually a very important constraint while doing structure optimizations. And the lowest energy for epsilon we can see is around 270 degrees, which is your option D. Then you have a question saying, you are given a 1D proton spectra of a nucleic acid in which peaks are present in the 3 to 15 ppm range. What conclusion can you draw from first look at the spectra? So this seems like a very difficult question, but it's actually very simple. If you remember the DNA versus RNA, the main difference is at this 2' position. DNA has 2 hydrogens in the 2' position and RNA has 1 OH in the 2' position. OH is an electron withdrawing group. Because of this, the 2' hydrogen of RNA gets more delta positive. It increases. So in RNA, the 2' hydrogen gets downfield shifted. So in DNA, the range of chemical shifts can vary from somewhere around 0.2 to 15. But in RNA, because of this downfield shifting of the 2' hydrogen, the range starts from around 3.5 or 3 ppm. So here the range is starting from 3 ppm, so it must be RNA. Because in DNA, the range will start from much more upfield values due to the absence of this OH. So the correct answer is option B, that is RNA, because the range given here is starting from much more downfield values, which is happening in RNA because of the presence of this OH. There is just one more question in the assignment 6, which is saying that sugar geometry is defined by what? So you can see sugar geometry is determined by many things. Firstly, you have this puckered amplitude. We have already discussed if the 2' is above the plane, it's called 2' endo. And if the 2' is below the plane, 3' is above the plane, it's 3' endo. But depending on how much it is above the plane, the geometries can be different. So we talked about a formula for finding this. So you have different torsions of the different bonds along the 5-membered ring. And the value of these torsions depend on 2 angles. One is the pseudo-rotation angle, which can vary from 0 to 360, and which is the angle that defines your this whether it's endo or exo. So the pseudo-rotation phase angle is important for determining the sugar geometry because it is the one which determines whether it's endo or whether it's C2' endo or C3' endo. The other is the tau m. Tau m is the maximum distortion from the plane of the ring. So if you consider this line as the plane of the ring, how much the 2' is above the plane is important. That is defined by tau m. You can also call this the puckered amplitude because it is the amplitude by which the 2' is puckering. So both A and C is important. So the correct answer is D, which is A and C. So why not the glycosidic torsion angle? So the glycosidic torsion angle, basically, I've shown this before here. Glycosidic torsion angle basically refers to the angle by which this so this is connected to the base, and glycosidic torsion angle refers to the angle along this bond. So it has nothing to do with the torsion, with the sugar geometry. It is basically how the relative orientation of the base with the sugar ring. That is why the glycosidic torsion angle has nothing to do with the sugar geometry itself. So that is why this option B is not considered. So your correct answer is D, that is both A and C. So that covers all the questions of assignment 6. As always, I'll upload the slides and the recording. I'll end the class here.